Hello, in this module we are going for clinical trial data analysis using R. It is a broad area, the application of R on data analysis on clinical trial. Generally, clinical trials conducted in phase 1, phase 2, phase 3 and phase 4 manners, where data from subject on humans comes from phase 2 and phase 3. Typically, in any clinical trial, parameter of interest, that is after giving some therapy to the patients, consider repeatedly over times, and that repeated observations comes through the follow-up study or follow-up periods visit of the patients. When we are having same patients repeated measurements over the study period, then there are some issues to consider for data analysis. On the other hand, when you are having more than one therapeutic group or arm, maybe 2, 3 and 4 arm, then issue become more complicated for the analysis and to find out the which treatment is doing better during the follow-up study. For example, we are having one existing drug to control the blood pressure, and we want to introduce another new drug also to reduce the blood pressure. Now we want to find out the new one is doing better control for blood pressures or not. For that, a typical clinical trial is required to perform, where patients' blood pressure data should be considered repeatedly over time, and the same drug to the same patient will be given repeatedly over time. There are so many challenges, but here we will put some lights on basic clinical trial data analysis using R. Let's start the module. Here this learning objective, here how to use R for competing risk analysis. Here we will illustrate one example through R. This reference is on competing risk analysis using R, an easy guide for clinician. This paper came on bone marrow transplantations on volume 40 in 2007. So it's an open source article. It is requested to you may go through this article also for details exploration and elaborations. So in these presentations we are using one function called cumulative incidence, that is taken from that from the author's website to run the competing risks. It is to be noted that in competing risks is one problem in high-level data analysis, where we are having problem with event. In generally, events defined as event happened or not, occurred or not, as if event happened is defined as 1, if it not happened it or censored case, then it is defined as 0. But sometimes, as we already mentioned, that event, that is death, may be not because of the therapeutic effect, may be because due to some other reason. For example, patient treated with cancer and they followed for certain period of time, maybe some years or some months, and after some follow-up, one patient or few patients died due to some reason like suicide, or due to some reason like some accident. So these are happened just because of death. We should not consider these death as event of our interest, that is considered as event due to the therapeutic effect. So because death reason is something different, so these factors should be considered separately. for analyzing data, and that can be done through cumulative incidence functions. So here, to analyzing competing risk event or competing risk modeling, cumulative incidence is one of the important functions should be considered for analyzing data. We will come later in these slides how to run with the cumulative incidence, but it is requested to go into details about the cumulative incidence function buildup in the author's website. Here, in addition to the cumulative incidence, we are also working with one library that is called CMRSK. This library is dedicated to handling the data with competing risk modeling, and as an example here we are considering first variable called DIS. DIS standing for disease type. So later on definitely we will define what is disease, different type of disease here, but here we are having two kind of disease. For example, one disease is defined as 0 coded disease, and another one is 1 coded disease. Second variable is f time, is basically the follow-up time. Here we are considering the follow-up time as days, but the follow-up time units maybe month or maybe year also. Here the third variable is considered as status. So at the last follow-up, the patient status has been considered. It is to be remembered that in earlier slides where we have done other survival analysis model, we have considered the status as 1 or 0, or sometimes we had considered 1 and 2. So 1 and 0 considered as their death happened or not happened. Again 1 or 2 considered as death happened or not happened. But here check, here status is given as 2, 1 and 0. So 3 variables are there. So where 0 stand for disease death not happened till the last follow-up. So these are the censored case. 1 stand for death happened, and death happened because of the therapeutic effect, therapeutic failure, and 2 stand for death happened but that those death were not due to the therapeutic failure. The reason of death were something else. So this is the status defining here. In this scenario, how to handle this data we will see. And this variable like disease, f time and status has been assigned to data.frame through functions of help as a database BMT. So BMT stands for bone marrow transplantations, and the data is considered from the bone marrow transplantation data. So we find easier to assign this variable as BMT. Now second step we are putting as str BMT. So it will give us the few values of this data frame assigned through the to the database BMT. It will give that we are having 3 variable with 35 observations. First is DIS having observation like 00001, and f time will give us the observations as how many patients, that is 35 patients observed in how many days. Here like 13 days, 1 day, 72 days of their observations. And third variable is the status variable, where it is coded as 012022200, like death happened. If it happened for other reason, then 2. If it death due to the therapeutic failure, then 1. Otherwise 0. Not now. We will go for next slide. As we mentioned earlier, that DIA standing as 0 and 1. So this DIA standing for 0 has been labeled as ALL, and 1's label as AML. So it's a type of disease presence in these bone marrow transplantations. It's a type of blood problem. So we will not go into details here, but now the status and disease type, that is ALL and AML, has been tabulated through the function called table within parenthesis DIS and status, and as a result it's coming that 0, 1, 2 for status as a first row, and as a column, first column disease, and after that ALL and AML coming, and how many were in AML with 0, it's coming as 2 persons were having censored in ALL and 9 patients were censored in AML. 3 patients were died in ALL and 6 patients died in AML, and 12 patients died in ALL due to some other reason, not for this therapeutic failure, and 3 patients died in AML due to some other reason, not for therapeutic failure. Now we will go for second slide. Now the mean values for follow-up time for both the treatment groups has been mentioned by T apply of F time and list of DIS and status with mean. So it shows that those were censored observations for ALL, they were having mean follow-up time was 53.50 days. Those who had died in ALL group, they were having 4.33 mean follow-up times, and those who are died but due to some other reason, their follow-up time was 9.33. In addition to that, those died in AML group, their follow-up time was mean follow-up time was 4.16. Those who are not died into AML group, their mean follow-up time was 32.55, and those who are died due to some other reason in AML group, their mean follow-up time was 6.33. It is required to be considered checked before directly applying the cumulative incidence, because if these measurement or mean values are not equally distributed in the both arm, then there is it can be noticed that there is a treatment failure in comparison to one arm to others. So here our main objective, how to find out that AML group is doing better in comparison to ALL group or not, and in place of where we are having problem with competing risks. Now come to the functions, that is cumulative incidence. This function is useful to deal with the competing risks, and this cumulative incidence within parenthesis if time, status, comma, DIS, comma, sense code is considered as 0. If you don't consider the sense code, by default it will consider the value is having the least value is having as a sensor code, but it is good practice if we can consider what is the sensor code. So here as a sensor code of this patient, censored has been defined as 0. So that is to define it, we had mentioned sense code equals to 0, and when we are talking on going for the making the plot, then we can go for X lab with a duration of month or days, anything. So now we are going to the result part, and these are results where it is tested the equality between these two groups. You see the estimate with different point of time is given like 0, 10, 20, 30, 40, 50, 60 and 70 days, and here also we in first column showing the ALL, AML, ALL and AML. So first column 1, that is those were died, for them the predicted survival probability is given by first row. For AML, for those are died, for them the predicted survival probability given by second row. For AL group, those died due to some other reason, that is what due to the competing risk, for them time wise like for 0 days, 10 days, 20 days, 30 days, 40 days, 50 days, 60 and 70 days, their duration of survival also been given. And for AML, those were in competing risks, for them also this corresponding survival probability given. The estimates observed as a standard error for the each time points also given next line, and for each time intervals. So now we are going for another application of the same functions. Again we are applying the cumulative incidence functions with F time, status, DIS, sensor code, and here we had segregated this time interval into 0, 3, 6, 9, 12, 26, 12, 24, 36 and 48 months or days. So whatever your unit you will prefer, you will put that, and here you can see that in that time intervals these estimates of survival probability, how Let's keep on changing. Corresponding for 1 stand for AL1 stand for those patients who had died and treated with AL group. AML1 stand for those patients treated with AML and died due to the treatment effect. Again AL2 here also standing for those patients treated with AL but died due to competing risks, and here 2 or AML2 also stand for those patients died due to competing risks. The importance of the competing risks is that it is very necessary to account the death due to other reason. For example, those patients treated with, here we are giving one example on BMT, but another example can be considered as head and neck, where this patient treated with radiotherapy, and those patients treated with radiotherapy, they are having some chance of die due to pneumonia aspirations. So as a effect of treatment, pneumonia aspirations occur and those patients die due to pneumonia aspirations, that can be considered as death as competing risks. So this is another example we can apply competing risk modeling in real life scenario. I'll come again into this result part. Again here in this slide we had modified our time intervals with different confidence interval as well, and we are getting the different results here. So earlier slide we was not having any level of significance, as by default we have considered 95% confidence interval, but here we may change our confidence interval for one level to another level, and we are getting the different results. Now again I am getting here the confidence interval as 0.95, and corresponding for L1 and L2 I am getting the results for lower and upper confidence limit, as the lower and upper are results I am getting. Again this function is applied for AL2 also, these functions to generate confidence interval for lower and upper. These are contributions. Now we are going for Kaplan-Meier estimations in presence of competing risks when we are having disease status, and we may apply by changing the disease status 2 as a 1, and we may check the results with the cumulative incidence observed as a competing risk, so we can find out the effect of death due to other reason into the sample data. That can be suitable to compare one side, consider the all variable as death or non-death without considering the competing risks, and apply simple Kaplan-Meier estimation procedure as a non-parametric choice, and in other hand check with competing risks that we have illustrated earlier and compare the results. So in that same data set we are going to apply Kaplan-Meier without considering the death status as competing risks or death. Now we are considering this data set as BMT1 and replace BMT as BMT1. Since we are having different code for status, again we are factorizing disease as ALN and AML, and we are preparing the table, and we are trying to find out the Kaplan-Meier estimations using the function called SART fit, and once we are putting this fit 1 as SART fit, we will get the survival fitting applications through considering the Kaplan-Meiers. Before that, again the distributions about the disease and status wise of follow-up measurements can be observed, like T apply value that we have seen earlier also. Coming the results obtained through the Kaplan-Meier estimation. Here disease 1 and 0, that is standing for ALL and AML, having confidence interval and maximum median intervals, median survival follow-ups, number of event, and results are coming here. So it is to be noted that here how the difference is there. Here in comparison to earlier results, here number of event is coming 15 for the ALL, and for AML it is coming only 9, where the median survival is obtained as 8 days, and for AML it is coming only 10 days. So with corresponding 95% confidence interval. So that is the difference results observed with considered with Kaplan-Meier estimator, but it is not suggested, just for illustrations and for exploration. It is requested to apply Kaplan-Meier estimation in addition to the application of competing cumulative incidence for competing risk modeling. But once you are having competing risk problem and you are aware that your data is having competing risk, then you should go with this function called cumulative incidence, because cumulative incidence function is the only applicable function that can solve the problem with competing risk modeling for survival data analysis. In this model, we have illustrated how to handle competing risk modeling using R. We have used the library called CRNPSK, and cumulative incidence function we have used to handle the competing risk modeling. I think it will help you for further problem with competing risk modeling in survival analysis.